The limitations of the central dogma, coupled with an explosion of new discoveries in molecular biology, paved the way for a new paradigm, the cellular dogma. Proposed by Stephen R. Quake in 2024, the cellular dogma recognizes the cell as a complex, interconnected network of information flow. This new dogma shifts the focus from a gene-centric view to a cell-centric perspective. It acknowledges that information flow within a cell is not limited to DNA, RNA, and proteins, but encompasses a diverse array of molecules and processes. The cellular dogma incorporates epigenetic modifications, non-coding RNAs, protein modifications, and cellular metabolites as integral players in the intricate web of cellular information. It recognizes that the cell is not merely a passive recipient of genetic instructions, but an active interpreter and responder to its environment. These modifications add another layer of complexity to the proteome, the entire set of proteins within a cell. Cellular metabolites, the small molecules that are the intermediates and products of metabolism, are no longer viewed as mere bystanders in the cellular symphony. They act as signaling molecules influencing gene expression, enzyme activity, and cellular responses to environmental cues. Section 5. A multidimensional flow information dynamics in the cellular dogma. Unlike the unidirectional flow proposed by the central dogma, the cellular dogma embraces the complexity of information flow within a cell. It recognizes that information flows in multiple directions, forming intricate feedback loops and regulatory networks. Gene expression is not solely determined by the DNA sequence, but is influenced by a multitude of factors, including epigenetic modifications, non-coding RNAs, and cellular metabolites. These factors can act as switches, amplifiers, or dampeners, fine-tuning gene expression in response to internal and external cues. The cellular dogma acknowledges the dynamic interplay between different cellular compartments. The nucleus, once considered the sole repository of genetic information, is now seen as one node in a complex network of communication that includes the cytoplasm, organelles, and the extracellular environment.